Hello, my name is Mrs. Tyler, and today we're going to do some GCSE biology revision where we're going to draw a knowledge organiser looking really at a group of things, how animals change over time. So we're going to be looking at selective breeding, genetic engineering, and then classification systems and how they've changed. This is on dual award and triple award. All you need is a piece of paper, you need it in landscape, so the long edge along the top. Let's get started. So like I said at the start, this is a collection really on how animals have changed or grouped over time. And it's got quite a long title, therefore. It's selective breeding, genetic engineering, and classification. And these are on both the triple and dual award GCSE. So everybody needs to know about these three things. How animals and ideas have changed over time. So we're going to divide the paper in half as usual, making sure that the long edge is at the top. This side is going to be on classification, grouping organisms together. This side is going to be on selective breeding, or artificial selection. And then slightly larger bottom half, it's not quite 2/3, about there. I'm going to do about genetic engineering here. Right, let's start with selective breeding then. Selective breeding is when humans select or choose the organisms that can reproduce, and therefore the characteristics are chosen for future generations. So it's when humans choose or select which organisms can reproduce, and it's based on the most desirable characteristics for humans. In exam questions, we often have to give the characteristics that humans might find desirable. So things like bigger, tastier, brightly coloured, fruit. And in terms of animals, it might be temperament. Okay, so you have to give the desirable characteristic. So basically, everything that we eat and all the animals that are domesticated, so that's farm animals and pet animals, have been selectively bred. And they've been selectively bred for a long time. So humans have chosen which ones are going to have babies, and then they choose those babies for many generations, changing the characteristics. An example of that then is wheat. So a long time ago, wheat would have had very, very small kernels of wheat on it. But due to random mutation, so in our video on evolution, we talk about random mutation, it might be that one wheat plant develops much bigger seeds. So humans then would just eat the small ones, but they'd save the seeds of the big ones for next year. So they'd eat the small ones, and they would save the seeds of these ones for next year. Which means next year, going to have a higher yield, but equally then you might have a variety of wheat that has big seeds, but also more of them. So you choose that one to save the seeds for next year, and so on and so on and so on. And so over the generations, the characteristics are changed. So it's also called artificial selection. Unlike natural selection, which is evolution, where nature decides which ones are best suited, humans decide which ones are best suited. And for example, this ear of corn here, the stem might snap because it's too heavy. So it wouldn't survive in nature, but humans have selected it. There are problems with selective breeding or artificial selection. The biggest one being that there is a reduction in the gene pool. So we've lost essentially the small genes to make the small wheat, which is fine for now, but we don't know if that one's got another advantage. Maybe it's more likely to survive disease. Maybe it's more likely to survive an increase in temperature, environmental change. So there is less variation, and that means that's inevitably a bad thing, because less likely to survive disease or environmental change. There's another problem with selective breeding in that it often leads to inbreeding. This is where individuals of the same family, so they will more likely to have the same genes, are bred together, and therefore they're more likely to get inherited recessive disorders. So in populations where there's inbreeding in plants and animals, you get an increase in recessive disorders. In genetic engineering, then, we're changing organisms, and again, we're choosing how we change them, but we're doing it by taking genes, sections of DNA, and putting them from one organism into another. So we're taking a gene, and remember, a gene is a bit of DNA that codes for a protein. So we're taking the code for a protein from one organism and putting it into a different one. So because it's got the gene, it's going to make the other organism's protein, which means it's going to have the characteristics that that protein gives. So that might be colour of fur, or making an enzyme, or making a certain protein. The example I'm going to give you then is for golden rice. Here's a section of DNA. It's actually from a carrot in this example. So it's carrot DNA. And carrots can make a molecule called beta-carotene. Right? And beta-carotene can reduce night blindness. It's needed to keep your eyes healthy. So scientists have found the beta-carotene gene and they cut it out using enzymes. Now, the problem that we've got is that we can't easily get genes from one organism into the other one. So we need to use something called a vector. A vector is just something that carries something else. So one type of vector that's often used is plasmid DNA, which is that circular DNA that can transfer between bacteria. So here's our plasmid, and we're going to stick, using enzymes again, our beta-carotene gene into there. So there's our carrot gene. So we've put it into a vector. In this case, our vector is a plasmid found in bacteria. Plasmids can get in and out of cells a bit easier. And so we can use that plasmid to get the carrot gene into a rice cell, and therefore into the rice DNA. So here's a section of rice DNA, and we're going to stick the carrot gene into the middle. So we do it to a single cell, and then we grow a rice plant from that single cell. And that rice plant will make, whenever it makes its normal rice genes, it will also make this particular carrot gene, which means the rice will end up a yellow colour. We call it golden rice because the rice will contain beta-carotene. So here's our rice. Rice has lots of small grains. But instead of being white rice, it will have a yellow golden colour, golden rice. So it contains the beta-carotene. So in populations where they don't have access to many fruit and vegetables, they might only have a carbohydrate-based diet, golden rice is going to give a better source of nutrition because it contains the beta-carotene. It's going to reduce night blindness. It's going to be better for their eyes. Other examples of genetic engineering are using bacteria to make human insulin to treat diabetes. So before we had genetically modified bacteria making human insulin, insulin had to come from pigs. It didn't work as well. Also, you had to have lots of pigs. It was quite a painful process for the pigs to get the insulin out. So bacteria that just make insulin is a much more cost-effective and actually a much better treatment of diabetes. The insulin's much better. We can also use it to make plants that are resistant to pests. So, for example, sweet corn, or maize, as it's called, can be made resistant to beetle larvae, maggots, because it contains a pesticide. So sweet corn resistant to pests. In this case, it contains a pesticide actually inside the cells that kills the maggots. Now that pesticide is at such a low level, it's dangerous to the maggots because they're so small, but it's harmless to humans. With genetic engineering, you could be asked an evaluate question. So you need to know the pros and the cons. So evaluate means giving both sides of the argument. So the pros of genetic engineering are that you can feed more people, you can increase yield, and you can make it more nutritious. You can treat diseases. And both of these are going to lead to a reduction in human suffering. The problems are, though, that especially in plants, these genes could escape. So because this carrot gene is in the rice DNA and therefore will be in the rice pollen that gets blown on the wind, that carrot gene could go and pollinate a different plant, and that gene could escape, which is fine in the case of carrot genes, but if it's in the case of a pesticide, for example, it's going to affect insect populations. So the genes could escape. So the new gene or the transferred gene could escape. In pollen. And affect wild types. So wild plants, but also wild insects. And also we don't know the long-term effects. Last one then is classification, which means putting organisms into groups. Originally, in the 1700s, it was done by using something called the binomial system. Bi means two, nomial means name, so it's the two-name system, which was developed by Carl Linnaeus, giving everything two Latin names. And it was Carl Linnaeus in the 1700s. And the two Latin names he gave everything were actually the two smallest groups of a system that he developed, grouping things into big groups and then smaller subgroups and smaller subgroups. And those groups were kingdom, phyla, so kingdom, phylum, class, order, family, genus, and species. So the binomial naming system just uses these two names. So Homo sapien, genus is, Homo is the genus and sapien is our species. So that's the binomial name. And kingdoms were the biggest groups, and these were phyla with smaller subgroups that sat within the kingdoms, and so on and so on. So the size of the groups gets smaller as you go down this list. You need to know these names. So mnemonics are quite common for this. So King Peter cried out, "For goodness sake," for example, would be a mnemonic. So that stayed right up until the 1990s. But then Carl Woese, who was American, another Carl, but an American scientist, decided that the kingdom of bacteria, the prokaryote kingdom, actually needed dividing up. So he changed it and he added a new level. Above that. So domain was added on much, much later. So Carl Woese said then that there should be, instead of just going to 5 kingdoms, the level above should separate out bacteria into 2 groups and everything else. So the 2 bacterial groups were called archaea and prokaryotes, or true bacteria, or modern bacteria. So archaea means primitive bacteria. And the reason he decided that they needed separating out from all the other bacteria is they've got really odd biochemistry, so often involving sulfur rather than oxygen, and they're often extremophiles, so they're found at very high temperatures or very high pH or very high pressure. So they're often extremophiles. And therefore shouldn't be grouped together with all the other bacteria because actually they are completely different. So prokaryote, as you know, means before the nucleus, and it just means true bacteria, so more modern bacteria. So he said if you divide the prokaryotes into archaea and prokaryote, then everything else is eukaryote. So all of the living things need to go under this domain. Eukaryote means organisms with a nucleus. And so essentially, it's the 4 remaining kingdoms. So there were 5 kingdoms in the old system, but there would be 4 eukaryotes. So it would be all the animals, plants, fungi, and the protists, which are the crazy parasites like malaria. And lastly, another idea, again that cropped up relatively recently, was showing evolutionary relationships through DNA analysis. So by sequencing DNA of all the different organisms, showed similarities. And we can draw evolutionary relationships in a diagram called a phylogeny, which shows a bit more detail because it shows when organisms became different. So I'm going to start a little bit there. I'm going to leave a bit of a gap. And here we've got our common ancestor. So in this, we're going to look at the evolution of different monkeys and apes. So it would be some sort of ape-like creature. So the first division that happened was the family that gave rise to the macaques. So in order of time, if these were in sets of 10 million years, then that would be 40 million years ago, the macaques branched off from the other apes. The other ancestor then carried on until about 10 million years later, where the orangutans branched off. Again, another 10 million years, and the gorillas branched off. And then lastly, about 10 million years ago, humans branched off and went on a separate path to chimpanzees. So this phylogeny, this evolutionary tree, tells us that humans and chimpanzees are most closely related, and humans and macaques are most distantly related. And even organisms like an orangutan, which is an orange-furred monkey, and a macaque could be an orange-furred monkey, you can see that it was 40 million years since they shared a common ancestor. So the macaques are most distantly related. All right, so that's our work on how organisms have changed over time, looking at selective breeding, genetic engineering, and classification. Your job now is to go away, reread that, and then have a go at some exam questions. Well done. and I'll see you next time.